All right, thanks, Logan. So before I get started, just a general announcement. You might have noticed that we're not doing questions at the end of each talk. We're going to save the questions for the panel discussion. So at the end, just before lunch, I'm going to ask all of this morning's speakers to come up to the front. We can use there's microphones in the room that we can use, but we can also use the audience response button in the app. So if you already have questions now, as the talks are going on, feel free to submit them in that audience response button in the app, and we can use that during the panel discussion to remember your questions. So with that done, what I'm going to be talking about builds on some of the talks we heard before the break about doing things on the surface, but this is now digging into how do we model cortical function, and I'm going to focus particularly on resting state functional MRI. I have no disclosures. So what we have once we've done all of this surface modeling and we have very accurately, very nicely registered, but really high dimensional and rich surface fMRI data. And usually the first thing that we do with that rich data is to summarize it into a lower dimensional set of features or metrics that we can then relate to behavior or cognition or any other individual difference aspect that you're interested in. And so when I'm talking in this particular talk about brain representations, what I mean is all of the analysis steps or analytical procedures that we take to reduce that rich fMRI surface data into a set of features for prediction or inference. And so my talk is set up into these three different sections. So first of all, I'm going to talk about what do we capture in a brain representation. Then I'm going to talk about who drives a brain representation, talking a little bit about group correspondence and individually specific brain representations. And then at the end, I'm going to talk a little bit about how to leverage multiple large data sets. So let's start off with section one. So this slide is just showing you a sub-selection of some of the available methods that are available for doing what we want to do with the brain representation, so extracting a set of features from that rich surface-based functional MRI data set that we have available. And this is not exhaustive. If your favorite isn't on here, I apologize. It's not personal. There's so many different options available. And that's partly because what we have in resting state functional MRI is just a very large data set, which is a signal processor stream. You can apply any signal processing tool that you want to this data set, and people have done that. And so unfortunately, in a half-an-hour talk, I won't be able to explain how each of these and all of the other ones approaches work. But I'd like to do is take a little bit more of a bird's eye perspective and talk about some of the aspects that these brain representations have in common or how they might differ. And so we can class these different approaches in different ways. So one of them is whether they provide spatial output. And in order to provide a spatial output, a brain representation would essentially have to estimate one or more features for every single vertex so that we can display that on the surface as we've seen in a lot of the different talks. And so all of the yellow ones here that I've highlighted can provide a spatial representation on the brain. Another common kind of type of feature that we can get from resting state functional MRI is a matrix representation. So the connectome here is the most common resting state functional MRI brain representation, which is a node-by-node matrix, where each element in this matrix represents the functional connectivity between those two brain regions. And so some more of these brain representations here can show you a matrix or a functional connectivity result. It just depends on there being one or more features estimated per pair of regions or parcels or areas or however we want to call it. Now, you might have noticed that some of these were yellow both on the previous slide for spatial representations and on this slide for matrix representations. And that's because some of these approaches provide multiple outputs. For example, independent component analysis, or ICA over here, estimates for each component or each resting state network a spatial map. And if combined with dual regression, you can also get a subject-specific time course for each of these networks or components. And then, of course, once we have a time course, we can do correlations between time courses to estimate a functional connectivity matrix. I should say that the PDFs of this should be available online, and I think each of these references on this slide and on all of the other slides are clickable, so that will take you straight through the paper if you find it hard to find otherwise. Another way in which brain representations differ is that whether they're static or dynamic. And so these ones highlighted here are static. It means that they still take the temporal fluctuations into account, but they estimate one kind of summary set of features, so one network map, for example, or one functional connectivity matrix that represents the mean over the entire scan. On the other hand, some of these brain representations are dynamic brain representations in that they explicitly try and model the dynamics over time. So for example, the periodic waves here is a movie, so you can see it's estimating a component that actually shows these traveling patterns over the cortex over time. Now, even within one of these, so the first choices that you have when you're trying to represent function or resting state functional MRI into a set of features is to select which of these or the many other options of brain representations out there you want to choose. But the next step that you have to do is even within one of these brain representations, there's quite a lot of choices. So I've already highlighted that this kind of connectome approach is one of the most common approaches to resting state functional MRI. Essentially, you select a set of nodes or brain regions, you extract the time course for each of those nodes, and then you estimate some pairwise similarity metric between each pair of nodes. But even within this brain representations, there's quite a lot of choices that we have to make. How do we define the nodes? What parcellation are we going to take? How many nodes are we going to use? How are we going to calculate the edges? And then even later on, how do we relate those edges to the research question of interest? And I think that's a topic that Ruby and some other talks this afternoon are going to talk more about. So with all this sea of options that I've laid out here now, one question that you might have is, well, do these brain representations actually differ? And these are some studies that have been done over the past few years that provide somewhat of an answer to this. The one on the left-hand side here is some work that I've done showing that between a matrix-based brain representation and a spatial brain representation, those we tend to think of as quite different and we tend to interpret as quite different, but in this work we showed that they actually share a relatively large proportion of variance, around 60+%. The next block over is some work from Gael Veracqua's lab looking at disease prediction for different parcellation choices. And at the top you can see the AAL and Harvard-Oxford, they perform a little bit less well. And we know that this is the case. We know that these are defined based on structural information, and Matt and others this morning have already told us that the functional boundaries do not necessarily map on well to the structural boundaries. But you can see between all of these functionally informed choices below that they all perform pretty similarly. And the same for some of Ruby's work. Again, you can see there are some differences, but in the grand scale there's not large differences in behavior prediction between these different brain representations. And then right at the end is again some more recent work from my lab. This is not a matrix of nodes by nodes, but this is a matrix of brain representations by brain representations. And again, you can see there's quite a lot of white, bright colors here showing that these brain representations don't differ particularly strongly in terms of the homology or the subject space that they're inducing. And so that means that these, even though there are so many different choices of brain representations, they share quite a lot in common. So is that lack of difference surprising? Well, if we remember that all of these are estimated from the same underlying data, it's not really that surprising that when it comes to the amount of variance that they explain or their prediction accuracy, that they actually share quite a lot in common. So why am I up here? Why should we care at all? Why does it matter? Well, I think it does matter for two key aspects. One of them is interpretability. You know, this is a question, where we differentiate between prediction and inference. If your goal is to get the most accurate prediction possible, maybe you don't care so much what's driving that prediction. However, if you're trying to make inferences, if you're trying to learn about the mechanisms of a disease or of an aspect of cognition, then you do care about the brain representations, because different brain representations are interpreted very differently. You know, how the spatial organization of a network is shaped, we think of that as a very different aspect of a brain mechanism than a temporal connection or a correlation between two brain regions. So that's one reason why I think we should still care. The second one is, I think we're all here to learn more about the brain, how the brain works. You know, I want to build on the work in my lab, but also the work of everyone else in this room to get inspired and to, you know, uncover the next step of the puzzle of how the brain is organized. And in order to get that kind of cumulative scientific advancement between all of us here, we have to be able to understand how a particular brain representation choice will impact the results, and how those results might show up if a different choice had been made. So I think that's also an important aspect for kind of making sure that we can understand kind of underlying mechanisms regardless of these choices. So I'm going to end each of the sections of my talk, each of the three sections of my talk, with the question of, okay, how do we navigate these sorts of challenges that I've just laid out? And I'll do that here for the multiplicity, or the kind of so many choices that are available for representing functional data. And these are just my kind of personal suggestions. I'd be happy to discuss this more in the panel discussion. But I think the first thing is to match the choice of brain representation to the research question. One bugbear that I have is that resting state data analysis is often described as exploratory, where it's really not. We have ideas about what we might find. We have knowledge about the population or the question that we're studying, and so thinking about that and thinking about what brain representation might best capture what you think might be going on can be very helpful. I think we can do more. I mean, we've done a lot of work in the field about test, retest, and reliability and generalizability. I think we can do more of that, apply more of that kind of rigorous attitudes towards comparing brain representations. And so my lab is starting a couple of efforts to do more benchmarking and more translation, and I hope to tell you more about those in the future. And then something that we've seen more and more is this question of underlying principles. So if all of these different representations of the same kind of rich, high-dimensional data have a lot in common, then maybe that tells us something about kind of the basic organizing principles of the brain, which could be interesting in and of themselves. And I think these three papers, again, they're clickable. They're very complex papers, so I don't have time to go into them in detail. But I think they are interesting and elegant examples of like trying to get at this idea of underlying principles. Okay. I don't think this is working very well, so I'm going to move on. So let's go to section 2, which is the question of what drives a brain representation. And here I want to talk about correspondence. So usually in most cases in neuroimaging, we want to compare brains from multiple people to each other. And in order to do that, we have to make sure that we're looking at the same region. So there's this tension between capturing individual brain organization as accurately as possible, but still being able to compare brains across different people and having the same thing to compare. And this can be challenging. For example, there might be missing regions that are there in some regions but absent in other regions. It might be these non-diffeomorphic splits, so a brain area that gets split up into two with another brain area kind of in between, which is very hard to align, as we've talked about a little bit this morning already. And maybe there's combined regions or kind of different dimensionalities that are optimal for representing one brain versus another brain. And again, this is this tendency between correspondence. So correspondence is important so that we're not comparing apples and oranges, where we have a reasonable comparison between individuals while still capturing the individual organization. So these are four kind of big picture approaches that people have taken to look at this individualization. And I'm looking at individualized nodes. So this is very much focused on the kind of boundaries or the spatial organization. I think Ruby this afternoon is going to talk more about sort of individual differences and how to relate that to behavior and other aspects. So I'll go very briefly over each of these and just kind of explain some methods and the advantages and disadvantages. So improved registration we've heard a lot about already. That's the easiest way to kind of put two participants in the same space. And MSMOL is one excellent approach that does that in brain space. So it uses both structural and functional information to align participants as well as possible. And we've seen the improvements in sharpness that can bring. Hyperalignment is another approach that people might have heard of, which does away with brain space altogether and tries and project individuals into a shared activity or connectivity space, which addresses these non-diffeomorphisms, but can be difficult to achieve and interpret. Separately, another approach to thinking about individualization is to just not care about the group level analysis altogether. So there's quite a lot of different groups and researchers nowadays that do precision mapping, which requires capturing a lot of data from an individual participant and then really focusing in and doing essentially a single subject analysis. So of course this is really good at capturing individual organization, but you can't compare as well to other data sets out there or other participants out there. The next option is to, first of all, get a group average result by combining all of our data together, and then afterwards map that group average organization onto individual subjects. And this is an elegant way of addressing the correspondence problem because we make sure that we start out by kind of a common solution for everyone that we then kind of try and make individual specific in a next subsequent step. And so that gives us correspondence across participants and comparability across the literature. But in some cases, these group priors can drive making up regions, can hallucinate regions that aren't there. And the fourth and final option that I'm going to cover here is this kind of integrated model. There are a couple of different examples here. So here the idea is, well, let's actually develop a hierarchical algorithm that knows that there are participants and there's a group level average, and that enforces a correspondence between them, but doesn't sort of estimate the group result first and then does the projection onto individuals separately. And so this can achieve both things. So it can enable correspondence across participants, often through weighted maps to avoid hard boundaries, and still do a very accurate job of individual subject solution, finding an individual solution by doing this iterative model optimization. Okay, so to wrap up section 2 of this talk, how do we navigate this challenge of individualization? there is going to be bias, and one step is to just avoid bias where possible. So these are a couple of things that we don't want to do. So when you're doing a data-driven definition of networks, for example, using independent components analysis, don't drive that by some subset, for example, only your controls, because that's going to introduce some bias, because the group average solution is going to look systematically more like your controls than your patients, for example. And also this post hoc correspondence, so driving a parcellation or a network definition from individuals only and then trying to kind of match them to each other post hoc can run this risk of apples to orange comparisons. There is still going to be bias. To some degree, you can capture information in the original data in multiple ways, and if you capture more of the individual differences spatially, then there might be less temporal information to look at, and vice versa. So there is a risk of kind of spreading out meaningful, behaviorally relevant information across pre-processing step, and that's just something to consider when you're interpreting the results. And one of the most useful tools we have for this section, and really for everything else, is to try and replicate and generalize results using either held out data or a completely new data set, and ideally even using different approaches. So with that, I'm going to go to the last section of this talk, and this kind of builds on what I've just said. So often we want to replicate, we want to generalize. More and more studies nowadays try to use multiple data sets, either from multiple labs or using some of the large-scale data sets that are available. And so this can be a challenge because sites differ even within one big data set, and certainly data sets differ in terms of the scanner platform, the sequences, and all sorts of different differences between data cohorts. And this is a problem because this can actually drive some of the results that we might find. So again, for this last slide, these are 4 different kind of philosophical almost approaches to working with multiple different data sets that I want to briefly highlight here. And again, I'm going to go over each of these in the next 4 slides and give you some advantages and disadvantages for each of these. So the first is harmonization, and this might be the most obvious thing that comes to your mind. This is essentially, okay, well, if we know that there are side effects, let's get rid of the side effects so that we can combine data into one large cohort. And combat is one of the most common actually algorithms to do that. There's multiple different versions of combat nowadays, and there's also some deep learning approaches coming out. And so this is an efficient approach, and it actually results into one large cohort for analysis. But it does only work on the summary measure. So essentially you're doing a different, you might introduce biases because you're doing a different combat for ICA, for example, compared to connectomic analysis. And it can also be difficult if your effective interests actually vary per site. So all of your patients were collected at one site versus another site. Now, this is a very good approach to use, and if you want to analyze all of the data in one go, this is really necessary and an important approach to use. The other approaches that I'm going to talk about really get away from this idea of analyzing, combining multiple large data sets into one big data set. So one option here that's pioneered by Thomas Yeo's lab is to do transfer learning. He called it meta-matching. And this is essentially the idea of training on one data set, a very large data set, and then using that pre-trained model and kind of adapting it into a newer data set. So this avoids the need for harmonization potentially because you're retraining the model anyway, and it even in his case avoids the need for exactly the same phenotypes because it uses the idea that phenotypes are correlated to kind of tweak a model and get better predictions, especially in smaller data sets. The third approach for kind of thinking about how to leverage multiple large data set in a way that doesn't require harmonization is to do a federated analysis. And this is leveraged a lot in the ENIGMA consortium, for example. Here, the idea is that you never actually have to share the data in the first place. You ask each of the different sites or owners of each of the different data sets to process the data in a way that you want it to be processed, and then they just give you the results from that analysis, and you combine those results in a meta-analysis or a mega-analysis. And so this avoids the need for data sharing. You can do this even when your ethics approval didn't allow for data sharing. It provides a higher level of privacy, but of course it isn't as open, and it might limit what you can do analysis-wise. And then the last and maybe thing that we should all really aspire to is true generalization. So here the idea is you train your model in one data set and you apply it to a different data set with the fixed parameters, without doing the kind of retraining or the tweaking that we've done before. And so this is, I think, what we all aspire to. You know, we want our results and our models to work in this way. It's the most rigorous test of generalizability and really the strongest answer to the replication crisis. But it can be pretty hard because these data sets are usually quite different in terms of their acquisition, their processing, even cohort differences. And so this is a challenging, but I think important aspiration to keep in mind as a field. So I'll wrap up this third and last section thinking about how to navigate multiple different data sets. I think one thing is to just plan for it, and there are a lot of open science tools out there like pre-registration and a registered report to decide beforehand what you're going to do, how you're going to use multiple data sets, and actually register your plan somewhere. And also realize that this using multiple data sets, even when they're freely available and freely shared, does still take quite a lot of resources and compute time and personnel time. And we're all interested in this idea of precision medicine, and I think we've made a huge amount of progress towards that by realizing and modeling and capturing individual differences. But if we're thinking about training a model that a clinician can use tomorrow anywhere in the world on his single one-subject data set, that seems still pretty challenging, and I think that's a useful thing to keep in mind as neuroimagers to try and move more and more towards that. So with that, I will thank the organizers of this educational course, Logan and Emma, and my lab. Some of them are here this week, so go and chat to them, and thank you all for your attention.